Okay, thank you all for being here. Can you hear me okay in the back? Okay, good. So, as Tony said, I'm interested in the problem of consciousness. I've been studying it for many, many years. And you see the title there. So, is your brain really a computer, as everybody's trying to tell you? Or is it something else? And I'm going to suggest a different idea, that it's more like a quantum orchestra tuned into the universe. Okay, so what do I mean by that? So we take it for granted that when we open our eyes, the world out there appears before us, or appears really inside our head. And being, the world appears out there. So being in this lecture will mean consciousness, awareness, as opposed to something else that the brain might be doing that's non-conscious. And it's approached through neuroscientists who probe the brain, roboticists, artists, physicists, psychiatrists, anesthesiologists, looks something like me for some reason, meditators, and philosophers. So I want to begin by asking a question. When did consciousness arise in the course of the universe? And so here we see the universe since the Big Bang, up until fairly recently. And so when did consciousness arise? Did it arise fairly recently with brains, with tool making and language, as many people will say? Or was it earlier with the advent of animal cells, eukaryotic animal cells? Or earlier with any life at all? Or did it appear even before the Big Bang? Now this has been proposed by Eastern philosophers and Colin McGinn, a Western philosopher, and also Sir Roger Penrose, who suggested that the Big Bang is actually preceded by another eon, which had its own Big Bang and goes all the way back, and that the universe is a serial and cyclical rather than parallel. So let's narrow this down and just ask the following question. Which came first, consciousness or life? Now neuroscience, psychology, most Western philosophy would say life preceded consciousness, life gave rise to consciousness, that consciousness emerged from living systems, from complex computation in the brain. On the other hand, panpsychists, Eastern philosophers, spiritually oriented people, Alfred North Whitehead, for example, would say that consciousness is intrinsic to the universe and therefore preceded life. Consciousness has been here all along. If that's the case, did life evolve to optimize consciousness? If consciousness was there before life, maybe life formed to take advantage, to access consciousness. In other words, did life evolve to feel good? And I think actually that's the case, as I'll try to tell you a little bit later. But first, the question is, what is consciousness? Now, in Western philosophy, this is supposed to be Plato. Plato realized that the world out there is all in our head. It's a representation. And it could be poorly representing, poorly portraying reality, as he said in his Plato's cave allegory, that these guys, troglodytes, were tied to these stakes and they could only see two dimensions. And for them, two dimensions was reality, and that was fine. They accepted that. It didn't occur to them that there might be more dimensions. And of course, people take this to mean that there might be more than the three dimensions we see, although I don't think that's the case, or four dimensions counting time. Descartes picked up on this idea and said that we could each be a mere brain in a vat, since we don't really know what's out there. We could be a mere brain in a vat, fed information by an evil genius. So here's a brain in the vat, bing, I'm walking outside in the sun. And that's presumably as real to that brain as our perceptions of reality are. So, and a lot of modern philosophers like Daniel Dennett would say that consciousness is actually an illusion. So we're left in this situation where both consciousness and reality, or one or the other, or maybe both, are illusions. So that's not very satisfying. I should also say that for Descartes, the observer was an immaterial soul which perceived the world in our head. Being is happening in this soul floating around. The soul is having an experience and connected to the brain through the pineal gland. Now that's all Western philosophy. On the other hand, in Eastern philosophy, consciousness pervades a deeper level of reality. Being is everywhere. Being is somehow intrinsic to the universe, an irreducible feature. And I'll come back to that point. So let's take up modern science, Western science. The brain is seen as a neuronal synaptic computer. Now really, the brain and how the mind works has always been compared to the contemporary information processing technology. The Greeks said that memory was a seal ring in wax. We had telegraph switching circuits. Freud said the subconscious bubbled up like a steam engine. Carl Friedman said the brain is a hologram. And then when computers came along, everybody said, aha, well, obviously the brain is a computer. And here we see a bunch of neurons and synapses here, and it's likened to a computer switching network with nodes and connections and variable strength synapses. Now, since that's thought to be the case, and since brain diseases and mental disorders of various sorts are a big problem, there's been a big effort, as you probably know, to map the brain, and called brain mapping, and governments around the world are pouring billions and billions of dollars and euros and yen and won and whatever into the idea that if we can map every neuron, every connection in the brain, and reproduce that in silicon, we'll capture the essential feature of the brain, including consciousness. We'll be able to build prosthetics. We'll be able to cure mental diseases, neurological diseases, Alzheimer's, and so forth. If we can only map all the connections of the brain, the neurons and connections, since they're just fundamental bits and switches, no different than in silicon. That's the premise. That's why they're asking and receiving these billions and billions of dollars to reproduce these connections in the brain. And you know, give us another 100 million or 100 billion, and we'll have it for you. That's the party line. However, it doesn't quite work. For example, there's this worm. It's called C. elegans, and about 15 years ago, they mapped every neuron, every synapse, every receptor and ion channel in the worm brain. It's 302 neurons and appropriate number of synapses. And they reproduced it. And so somebody said a couple of years ago, well, wait a second, before we worry about a human brain or a mouse, let's try the worm. And so they recreated the worm brain in a computer, or many, many computers. It's a worldwide effort. And had a big rollout announcement, such and such a day they were going to turn it on and stimulate this virtual worm and see what it did. Well, the day came and went, and nothing happened. The worm just lay there. It didn't respond. Something's missing. So a simulated worm, and normally the worm would swim around and do all kinds of things. So with its 302 neurons and thousands of synaptic connections, it didn't work. Now, on the other hand, consider a single cell organism, a living organism, a paramecium. It swims around without synapses. If it bumps into something, it backs up, it goes the other way. It finds food, it avoids predators, it avoids obstacles. It can learn if you suck it into a capillary tube, it gets out faster and faster each time. It can find a mate and have sex. And there is a X-rated picture on the right of two paramecium fused during sex. It's the only time they're absolutely still, strangely enough. And we don't know if they're conscious during sex, but if they're going to be conscious at any time, that'd probably be a good time. And yet it doesn't have any synapses. It's one cell. There's no connections. So if a paramecium could be that clever, is a neuron as dumb as being simply on or off, or an integrate and fire device? So that's the problem. Now let's look at the paramecium a little bit more. How does the paramecium do it? How is the paramecium so clever? Well, it uses structures over here that stick out from its surface that are called cilia, that are both sensors and motors. So if it bumps into something, the cilia bends, and it sends a signal back in the body of the paramecium, and it makes the other cilia move in a coordinated fashion, like oars, and it gets the hell out of there, and it swims away quite nimbly, or it goes finds a girlfriend or a boyfriend or whatever. And these cilia are, if you look at them, these are found widely in biology, in pretty much all animal cells, including ours, in our lungs, in our GI tract, in all of our cells have a very similar structure called the centriole, that are 9 doublets or triplets of structures called microtubules that I'm going to tell you about in a little bit. So here's a cross-section of cilium coming out of a paramecium. Here's a cross-section of the cilium itself. It's got 9 of these cylindrical tubes, microtubules, and here's a centriole, which is responsible for cell division. And this is what got me interested in microtubules 45 years ago in medical school, studying mitosis. And okay, so why am I telling you about cilia in a paramecium? Well, I'll get to that in a second, but how the paramecium bends is very interesting. So here's a cross-section of a paramecium. You see these microtubules, and here's one of them. And they have these motor proteins called dynein, and the dynein uses ATP to contract. And so in a very coordinated fashion, all the dyneins contract at the same time, and the cilia bends in a coordinated fashion. Not just one cilium, but all the cilium. They work in a coordinated, concerted fashion to perform the functions of the paramecium. Now, it turns out the same mechanism, the same microtubule, and the same motor proteins are found in all of our neurons. So jump ahead billions of years in evolution, look inside our own brains. And here's a neuron, and this is the cell body, this is the axon, and we're going to zoom inside one dendrite. So we know that if a synapse is out here or somewhere, to learn, to remember, you have to upregulate the synapse or downregulate the synapse. And to do that, you have to transport materials from the cell body out to a synapse here or here. And that is accomplished by these same motor proteins, dynein and another one, kinesin, which transport material to a specific place on the neuron, has to jump from microtubule to microtubule, has to go left or right at every branch point, and get to the right synapse. And these dyneins and kinesins carry their cargo. And how do they know where to get off? What's their guidance mechanism? It turns out that this protein called tau, and you may have heard of tau from Alzheimer's disease. Tau is a microtubule-associated protein that is deposited at certain specific locations along the microtubule lattice. And it's kind of a code. It's kind of a traffic signal. And it's telling this kinesin to get off here and deliver your cargo. It's kind of like a FedEx code or something like that. It's coded into the microtubule, and it tells the motor proteins where to deliver their cargo. So this is really memory, because if you're upregulating synapses, you need to deliver the cargo. And this tau, we also know that in Alzheimer's disease, the tau, shown here in red, falls off, and the microtubule destabilizes. And when the microtubules destabilize, you get Alzheimer's disease. Now, whether the tau falls off first, and then the microtubule destabilizes, or the microtubule destabilizes, then the tau falls off, is a little unclear. But in either case, we lose the integrity of the microtubules. So in Alzheimer's disease, so here's a diseased Alzheimer's neuron, and it's kind of crinkly because it's lost its shape because its microtubules are messed up. And if we look inside, the tau, shown here in blue, has fallen off, and the microtubules, subunits, fall apart. So we lose the microtubules, we get Alzheimer's disease. And the symptoms of Alzheimer's, now we know about the amyloid plaques that are outside the cell, outside the neuron, but the symptoms, cognitive dysfunction, occurs when the microtubules fall apart and form these tau fall, neurofibrillary tangles. Now I'll come back to microtubules in a few minutes, I promise. I've been obsessed with microtubules for 45 years, and maybe you'll see why by the end of this lecture. So let's go back to Plato and what's happening in his brain, or our brain, as your brain, as you're listening to me and watching the slides. So Plato has the light come in, and he has this bing of the tree, and the same thing's happening with you with the slide and looking at me. And information comes into the thalamus. In this case, visual information comes into the thalamus. And it takes 3 steps, 3 waves for consciousness to occur. The first step in vision is to the back of the brain, V1. And then there's a second wave forward, associate to the front of the brain, the second wave. And then from the front of the brain around prefrontal cortex, there's a third wave. And it's the third wave that seems to correlate with consciousness. So this is why it takes about 300 milliseconds to have conscious perception in vision. Now, this is actually consistent with a philosophical theory of consciousness called higher order thought, put forth by David Rosenthal and Rocco Gennaro, where anything, in order to be conscious, has to be projected from the prefrontal cortex, somewhere else in the cortex. And it's also physiologically known that it's the third wave that correlates with consciousness from the work of Victor Lame. And also, it's been shown by George Mashur and others that it's the third wave, and only the third wave, that's affected by anesthesia. Now, this is really strange because the neurotransmitters and the neural function in waves 1 and 2 are pretty much the same. But it's only the third wave that is conscious, and only the third wave that's affected by anesthesia. All types of anesthesia, gas anesthesia, propofol, and even ketamine affects only the third wave. So what happens here? Well, when this wave gets into the cortex, there's also 3 waves. So here's the cortex, and here's thalamic inputs coming, or associative second or third wave coming in to layer 4. From layer 4, signals are sent to 1, 2, 3, and 6. and 1, 2, 3, and 6 converge on layer 5 on these giant pyramidal cells. And these giant pyramidal cells are what are responsible for EEG, because their apical dendrites go vertically up to the surface of the cortex, and everything else cancels out, and they give rise to EEG. And so it seems that this third way, the giant pyramidal cells are somehow most directly responsible for consciousness. That's not saying it's not happening elsewhere, but if it's going to be happening anywhere, it's going to be happening in these giant pyramidal cells. So let's go a little bit deeper. If we look inside these giant pyramidal cells, we see microtubules, the same structures I showed you before. Now, in dendrites and soma, interestingly, they're in these mixed polarity networks. They're not continuous. Now, they're part of the cytoskeleton, which is thought to be structural support. But, you know, like our bones are structural support, our skeleton. But if you want a structural support, like in your femur and your thigh bone, you wouldn't break it into pieces and have it side by side. That doesn't make any sense. It's not structural. So these have been shown through simulation to be this arrangement optimal for information processing, not structural support. So, and here's the structure of microtubules showing different states. So it looks like consciousness is happening here in these, in the dendrites and soma of pyramidal cells, where we have these mixed polarity networks, which are unique in all of biology. In axons, the microtubules are continuous and the same polarity. In the dendrites and soma, they're broken, interrupted, and a mixed polarity, only in dendrites and soma. Anesthesia acts on dendrites. It looks like consciousness comes from dendrites, and I will argue memory occurs in dendrites. So let's talk a little bit about memory, because I'm going to suggest that memory is stored in microtubules. Now, here's a model of memory called long-term potentiation. It's standard neuroscience for many, many years, where you have an axon and a dendrite, and if you stimulate the axon high frequency for a short period, high intensity short period, you get a prolonged sensitivity at this synapse. This synapse becomes overly sensitive. It takes less to stimulate it. And that's the synaptic plasticity, and that's thought to be the mechanism for memory. However, these proteins are transient, only last hours to days, yet memories can last lifetimes, as we know. So memory has to be stored at another level inside the neuron. So what might that be? Well, one of the things that happens in memory is that this enzyme, CAMK2, gets activated, and it phosphorylates something in the neuron, which can then store memory for a long time. So what might that be? Well, here's this enzyme CAMK2. When I first saw this, I thought it was a joke, because it's such a beautiful molecule, looks like a snowflake, and when it's activated by calcium from the neurotransmitter, it extends these arms or legs or whatever you want to call them, kinase domains they're called, upwards and up and down, so it kind of looks like a nanopoodle, people have called it. It's nanoscale, kind of like a French poodle if you want to use your imagination. And over here we see the phosphorylation sites where it can deposit phosphate energy and store energy for a long period of time. So what is being phosphorylated inside the neuron by this molecule? When I saw this, I said, hexagonal. Well, microtubules have hexagonal lattices. And so I got with my colleagues, and we decided to model and simulate CAMK2 and microtubule lattices. So here in the gray, we have two types of microtubule lattices. And here we have the CAMK2 overlaid, and we see that it perfectly matches. So what we proposed in this paper in 2012 was that the CAMK2 nanopoodle, if you will, looks kind of like a lunar lander. If some of you remember the moon landing some years ago. It comes down on a microtubule and binds up to all 6 of its kinase domains, and can phosphorylate them and leave a memory trace. So this is what we think is happening in memory. The CAMK2 lands and leaves this memory trace, which can then code for tau or anything else. And and it can be hardwired, and that's long-term storage of information and memory, rather than the synapse, which is very transient. And this means that there's a ton of information processing in microtubules. The capacity, even for a small amount, is enormous. And when you add up all the microtubules and all in one neuron, it's enormous. I should also say that the centriole that I showed you earlier seems to store epigenetic memory. We know that when I learned genetics in medical school, it was like only genetic information could be conveyed from generation to generation, but now we know there's a lot of epigenetics. If you learn a behavior, it can be transferred to your offspring, and this has been shown over and over again, and this seems to be the mechanism that the epigenetic memory is stored in the centrioles, which are part of the cell cytoskeleton. So all this suggests that microtubules are processing information. Now in the early, I got interested in microtubules in the 70s and started writing about them, and in the 80s did a bunch of simulation with people where we modeled a microtubule, and it's made of these individual proteins called tubulins, which we imagine could be in 2 different states. Turns out it's more complicated than that, governed by some electron function, and modeled it as something called the game of life, which is a simple cellular automata where you just have local neighbor interactions and you get information processing through the microtubule. So here's just an example of simulation showing information processing going through the microtubule. And I published a bunch of papers and a book about this, information processing in microtubules, and later got into quantum computing in microtubules with Roger Penrose that I'll talk about. And I should say it's similar to a language structure that Massimo Palmerini-Piatelli, as some of you may know, and Chomsky have talked about, that this could be also language encoded. But the point is this, that the brain mappers, people who want you to give them billions of dollars to map the brain, are saying, here's all we got to do. So that includes artificial intelligence people, singularity people, that there's 10 to the 11th neurons per brain, 1,000 synapses per neuron, operating at about 100 hertz, gives you 10 to the 16th operations per second per brain. So just give them another couple billion, they'll build a computer with 10 to the 16th operations per second for the computer, and you'll be able to put your consciousness into it when your body is about to give out. All it needs is a couple billion. Well, we came along and we said, no, that's naive. Consider the paramecium. Or consider a neuron in a brain. There's 10 to the 11th neurons per brain, 10 to the 9th tubulins per neuron, 10 to the 7th hertz, because they're oscillating at 10 megahertz. And if you look at one neuron, you get 10 to the 9th and 10 to the 7th is 10 to the 16th operations per second per neuron, as much as what the AI singularity people are saying for the entire brain. And you would need 10 to the 27th operations per second per brain. Well, we got very unpopular with AI people who were kind of stealing their lunch. And they said, go away, you're bothering us. And they launched a counteroffensive and blah, blah, blah. It's a long story. But one day somebody said, I was going around kind of spoiling the party, and somebody said, well, let's say you're right. How would that explain consciousness? And how would that explain love, emotion, fear, feelings, all that kind of stuff? And I had to admit that I didn't have an answer. It was just more computation, more reductionism. Where's consciousness? Where's the being, if you will? Is something else required? And could it be quantum physics? Well, fortunately, the same individual told me to read a book, suggested I read a book by Roger Penrose, now Sir Roger Penrose, called The Emperor's New Mind, which I did. And this book really changed my life. It was kind of over my head, but I tried to absorb as much as I could. It was many things. First, it kind of trashed AI and said consciousness is not a computation. And this was a long half of the book, but the basic idea is this. A computer can do a lot of things. It can beat you in chess, it can do all kinds of things, but it doesn't understand anything. Now, John Searle made the same point with his Chinese room argument, and that you can sort Chinese characters, but you don't really understand Chinese. And Roger said, understanding is not a computation. They need something outside the system. You need something non-computable, he said. And that something was a quantum physics mechanism for collapse of the wave function. Now, I read this, I was kind of stunned, because I hadn't really thought about quantum mechanics very much. And it seemed to stretch. He's going from the brain to quantum physics, whatever that was. But he seemed like a really bright guy, and it was the only actual mechanism for consciousness I had ever heard. And that remains the case. Nobody else has a mechanism for consciousness. It's said to be, you get enough computation or enough complexity and consciousness happens. But those are all really hand-waving arguments. There's no threshold. There's no real theory behind it. So it forced me to try to delve into quantum physics. And as Richard Feynman said, anybody who claims to understand quantum physics is either lying or crazy. Because it's so bizarre, so weird, that nobody really does. So I'm going to give you a very brief version. The world, it seems, is divided into two realms, the quantum world and the classical world, the classical realm. Now, in the classical realm, everything is fairly predictable, follows Newton's laws of motion, the Maxwell's equations, the gas laws, gravity, et cetera, et cetera. And that's what we're familiar with. That's where we live, basically. On the other hand, at very small scales, and we don't really know the cutoff. We don't know how small you need to be to go into the quantum world. And the level seems to change. You have quantum superposition, where things can be in multiple places at the same time, smeared out like a wave, or in two or more states or places at the same time. Things can be, quantum particles are going to be separated and remain entangled, so if you mess with this one over here reacts, and they can condense into unitary objects, quantum coherence or condensation. So this leads to a lot of questions, like, for example, why don't we see quantum superpositions in the classical world? Why don't we see things smeared out all over the place? Why do we see things in only one definite place? Where we know from experiments that if you're not looking or you're not measuring them, they can be in multiple places. Well, the early pioneers of quantum mechanics noticed this, Niels Bohr, and it turns out that if a machine made a measurement on a quantum system, it appeared as though even in the machine the results can remain in multiple possibilities until someone looked at the result, the human observer, the observer effect in quantum mechanics. And this became known as the Copenhagen interpretation because Niels Bohr was Danish. And it seemed to work. It allowed them to go on and do experiments, and they did a lot of good stuff, but it kind of pushed the underlying problem under the rug. You know, what was reality really like? How could things be in multiple places? So Erwin Schrödinger came along, and he didn't like the idea. He thought it was bizarre, which it is. And so he came up with this famous thought experiment called Schrödinger's cat, where he coupled the fate of a cat to a quantum event, say outside this box, so that a quantum particle would both enter the box and not enter the box. It would both release the poison, not release the poison. It would kill the cat and not kill the cat. Until, he said, according to Copenhagen, someone opened the box, took a look, and only then would the cat be either dead or alive. And this was intended to show how ridiculous this conscious observer was, but nobody's really come up with a proven alternative. So it remains a very viable option, that consciousness causes collapse of the wave function. There's also the double slit experiment that seems to support that anyway. Now there's some other ideas like decoherence, that any interaction between a quantum superposition and classical world disrupts the quantum state, but it doesn't really get rid of the quantum state, it just kind of mixes it. And how can any quantum state be truly isolated? Because at the quantum level, there's no classical world, it's interacting with other quantum states. Nonetheless, it's still out there. Multiple worlds. This is actually very popular. It says that superpositions are actually separations in reality. So if a particle is in two places at the same time, the underlying reality, space-time geometry, separates. And there's no collapse. Each possibility branches off to form a whole new universe. So every superposition evolves its own universe, and we have an infinite number of overlapping universes, multiple worlds, and or parallel universes. And as bizarre as this is, it's actually the most popular interpretation because it gets rid of a lot of problems. It introduces a lot of problems, but the math works and people are happy with it. It's bizarre, but it seems to work. Mathematically, I don't believe it myself. But anyway. Then there's some others, and then Roger Penrose came along and formulated this idea in that book, The Emperor's New Mind, where he said instead of consciousness causing collapse, which is the Copenhagen interpretation, which by the way puts consciousness outside of science, it's some mysterious thing that causes collapse, he said the opposite, collapse causes consciousness. But it has to be a self-collapse, it's going to happen on its own. He also said, as I mentioned, that understanding is more than computation, and involves a self-collapse of the quantum wave function. And he agreed with multiple worlds that superposition is a separation in underlying reality. Underlying reality actually begins to separate or bifurcate, but that these separations are unstable and will reduce or collapse by an objective threshold given by the uncertainty principle E equals h over t, where E is the amount of superposition, h is Planck's constant, and t is the time at which collapse will occur. So each, and you call this objective reduction. Reduction or collapse of the wave function by an objective threshold, and OR. So each such OR event is accompanied by a subjective experience, Bing. So he introduced consciousness into the universe by self-collapse. And moreover, this is a real kicker, that the choices were not random, as they would be in Copenhagen or decoherence, but would be influenced by what he called Platonic values embedded in space-time geometry, embedded in the structure of the universe. So he essentially was connecting consciousness to the structure of the universe. Okay, so let me back up, because he also took the question of superposition seriously and asked the question, how can particles exist in superposition in two or more locations at the same time? The answer, he said, for the answer, he turned to Einstein's principle of general relativity, where mass is equivalent to curvature in space-time geometry. So he had these cartoons in his book, and he's taken 3 dimensions of space and made 1 dimension of space and 1 dimension of time. So we've gone from 4-dimensional space-time to 2-dimensional sheets, so we can put it on a slide and make drawings and look at it. So the particle over here would be a curvature in this particular location. Now a particle over here, let's say it moved, would be here. So you have 2 different curvatures. Now in that case, superposition is actually 2 different curvatures in the particles in both places at the same time. Now if you believe in the multiple worlds, then that the superposition separation would continue and you get 2 universes. There's a particle in one universe, a particle in another universe, then that'll happen again and again and again, and you get an infinite number of universes. So that's the multiple worlds. However, Roger said no, at a certain time, t, given by E sub g, the amount of superposition, there will be a self-collapse. This one will disappear, this one will continue, and this is the threshold for objective reduction, and a moment of consciousness occurs at that moment. Now, if this is occurring all around us in the table and the air, there would be these random, spurious moments of consciousness. This seems odd, but a lot of people are turning to panpsychism now, that consciousness is attributed to matter or whatever. But to get the kind of consciousness we have, it needs to be organized in a certain way, and I'll come to that later. But when it does become organized, what this implies is that consciousness, conscious experience, is not necessarily a pattern in her brain, although it correlates with a pattern in her brain. It correlates with a particular pattern in space-time geometry. This is the essence of the redness of the rose, Bing, which reproduces the same pattern of space-time geometry in her brain. And where in the brain we'll come to in a second. So when I read this book in the 90s, I thought, he's got a great mechanism for consciousness. But he doesn't have a biological component to do it. He doesn't have a quantum computer in the brain that could terminate by this OR mechanism. And I said, microtubules are what he is. I've got the structure, he's got the mechanism. So I wrote to Roger, and I sent him some articles I'd written, pictures of microtubules, and we soon met in his office in Oxford and began to collaborate, and I invited him to the first Tucson conference here in 1994, after which we went to the Grand Canyon. There's Roger Penrose, there's me about to fall in. That's Dave Chalmers, some of you may know him, and some other, Roger's wife. And we started collaborating on a theory which first published in '95. It was immediately attacked, ridiculed. They tried to get rid of it before it even was published. The brain's too warm, wet, noisy. I'll come to all that. But it has survived, and actually, I would argue, is getting stronger all the time. So we called it Orchestrated Objective Reduction, OR, Objective Reduction from Roger's mechanism. Orchestration has to do with what happens in the microtubules inside the neuron. So the basic idea is that inside neurons we have individual tubulins that can be our protein qubits, quantum bits that can do quantum computing, and when they reach a certain threshold, they self-collapse with a moment of Bing. And here we are a couple years ago in where was that? Stockholm. And he was like a rock star, and I was trying to protect him as kind of his bodyguard there. But this idea that the protein changes shape, and I think Tony mentioned this, conformation, turned out to be wrong. And because that would require energy and generate heat. We don't want that. That would mess up the quantum state. So we've actually changed that. And now it's strictly electronic dipoles, although it has to be coupled to the mass of the atomic nuclei. So then I began working with Travis Craddock and Jack Jasinski, looking at the structure of tubulin and the microtubules, and in it are these aromatic amino acids, tryptophan, phenylalanine, and tyrosine, which have these pi resonance clouds, which are quantum structures. And here you see where anesthetics act in the microtubules. And so let me talk briefly about these pi resonance clouds, because they're the basis of organic chemistry, and also for consciousness, I would argue. And I'll show you why in a second. So back in the 1700s, chemists were working on hydrocarbons, and they knew about alkanes and alkenes that were chains that followed the formula. You can see there CnH2n plus 2 or CnH2n. But then they had benzene, C6H6. Now they didn't show the structure. I'm showing you the structure there, although it's actually missing an extra bond there. They knew C6H6 was oily, flammable, smelled funny, but they didn't know its structure. And then one day, Kekulé had a dream that these alkanes and alkenes were like snakes, and one snake swallowed its tail, and it was a ring, and he woke up and said, benzene's a ring. And he was right. And benzene is indeed a ring, and it's not only fuels our cars and whatnot. In bulk form, it's flammable, but it's the basic component of organic chemistry in biology. We're basically built on these types of rings. Carbon chemistry in general, in particular, these rings. Now, why are they so important? Well, you see these three bonds, and these three bonds, extra bonds, can rotate or can switch, or actually can go into quantum superposition. So these benzene or phenyl rings, they're called when they're attached to something, they share three delocalized electrons, and they form pi resonance clouds, where the electrons are smeared out over the whole ring. And this is the quantum effect. So they're not in any one place, they're kind of everywhere at the same time. And anesthetics act in this type of environment. I'll come back to that in a second. Now, these pi resonance clouds are important in consciousness because anesthesia acts where they are in the brain, and psychedelics and psychoactive drugs depend very much on these molecules. For example, dopamine, the pleasure molecule, is based on this ring with a non-local pi resonance cloud. Serotonin, LSD has a complex ring, DMT, psilocybin, they're all these pi resonance clouds that actually, it was shown in the 70s, promote quantum resonance. When they bind to something, they push quantum resonance into their receptor. So the receptor, whatever it is, becomes more quantum, becomes more in the quantum realm. Now, if you go the opposite way, look at anesthesia, which takes it away, these guys, Meyer and Overton at the turn of the, around 1900, both show that all anesthetics, and all these gases are anesthetic, nitrogen at several atmospheres, and methoxyfluorine, a very potent anesthetic, cause anesthesia in all animals and at the same concentration. It takes the same amount of anesthetic gas pressure to put you to sleep as it does to put a salamander or a dog or a mouse or a fruit fly or anything, the same partial pressure. And their potency, so they took all these gases, anesthetics, and they tried to find one common parameter because their chemical structure is completely different. And they found that they lined up perfectly along this line with solubility in olive oil, which is basically the pi resonance benzene molecule I showed before. So the more soluble they were in that type of medium, the more potent they were in putting people to sleep, or animals to sleep. So if you look at a human body, or any body, there's all these solubility phases, and polar would be out here, like in the blood and water and charges, and down here the non-polar benzene right here. So that's where consciousness comes from in the brain. If you take away the quantum effects with anesthesia, you become unconscious. If you jack it up with psychedelics or dopamine, you get more conscious. So I won't have time to show you about anesthesia, but people assume that act in membrane proteins. The latest data is they act on microtubules, and it looks like they act right here in these channels. So, and Meyer and Overton were the guys who invented that. So I mentioned that in our early days of our theory, we had the protein changing shape and being a superposition of both states. But we've changed in around 2002 to having oscillating dipoles. The protein doesn't really have to change shape, it just has to change state from the dipole going up versus the dipole going down. This doesn't require a lot of energy, it's very low energy, and it can go into superposition of both states and be a quantum bit or a qubit. So the theory now has evolved to where if you take microtubule inside Pyramidal cell, for example, has different states. If you take one little row of tubulins, you have a dipole going up, dipole going down. Dipole can be in both directions, a superposition that's not shown here. And you bring in an anesthetic, and it stops the oscillation, and you lose consciousness. So that seems to be, and the latest results from the University of Pennsylvania seem to show that anesthesia works on microtubules in this way. So basically the theory we suggested was that superpositions, E sub g, grow and evolve till we reach this threshold at time t, and you get bing, you get a conscious moment, and then it starts all over again. And the choices are influenced by those platonic values I mentioned. I'll come back to that. So if we relate it back to the space-time geometry business, as I said before, everything is curvature in space-time geometry, the structure of the universe, and bing occurs when we reach this threshold, and there's a collapse, and we choose a particular state. So in the brain, we have a series of these bing moments. Here, 25 milliseconds at 40 hertz, but it's probably much, much faster, and I'll talk about that in a second. So every collapse occurs, and you have a moment of consciousness, and it sends information both forward and backwards. There's backward time effects in the brain, and that's been shown experimentally. And in fact, without that, we wouldn't be able to act in real time and have free will. Okay, so it's a beautiful theory, and then reality set in. Well, not reality, but criticism set in. For example, this guy, Max Tegmark, in 2000, well, everybody was saying, well, the brain's too warm, wet, and noisy for these quantum effects. Because if you go to a laboratory and you want to build a quantum computer, like with ion traps or this or that, any temperature vibration will mess it up. So they said, you know, they have to do it at absolute zero. So how could this happen at 37 degrees, for example? And we said, well, you know, biology has had billions of years to figure this out. You know, there's mechanisms, we propose mechanisms. And this guy, Max Tegmark, published a paper in 2000 where he calculated the decoherence time of a microtubule. He said he had temperature in the numerator, which was kind of weird. But the mistake he made was having the superposition separation 7 orders of magnitude too big. And we wrote a paper a year later in the same journal and corrected it, and we got a superposition time of about 10 to the minus third, 10 to the minus fourth, maybe slower or longer. But that was the calculation. We really wanted 25 milliseconds, but it didn't quite get there. But it's certainly not 10 to the minus 13. It turns out 10 to the minus fourth is probably enough. And then more evidence came along that actually temperature is not the enemy of quantum effects. And for example, this study showed that quantum spin transfer through these benzene rings is actually increased by temperature. That it vibrates incoherently, and you get more quantum effects as you raise the temperature. And then finally in 2006 and so, there began a series of papers showing that plants utilize quantum coherence in photosynthesis. So what happens in plants, they collect light, and the photon energy is carried over to this reaction center to make food, and it goes through this protein through a series of chromophores, which are the same pi resonance clouds I was showing before. And it goes through all these chromophores simultaneously. The energy occupies all pathways at the same time, a quantum superposition. And that gives a very efficient transfer. That's why we get the food we get. Otherwise, you know, we might not be here. We might not, animals wouldn't have vegetables to eat. We wouldn't have vegetables to eat. We wouldn't have the animals to eat. So this highly efficient process, which is now being applied to photonics and solar cells and so forth, works in plants. So if a carrot or a potato or a rutabaga can utilize quantum coherence, it's likely that our brains could do so. And in fact, in 2000 well, I met this guy in 2009. He published in 2013, 2014. His name's Anirban Bandyapati. He'll be at our conference. He's quite an interesting character. He liked our theory, and he's an amazing experimentalist. So he took a single microtubule, shown here in the red, and he applied 4 electrodes, 2 to stimulate, 2 to record. And if you start with, the microtubule is a good insulator, but if you stimulate at certain frequencies, and look at the conductance, at certain resonant frequencies, it becomes highly conductive. The resistance drops. For example, 10 megahertz, 20 megahertz, 10 kilohertz, also gigahertz. So at those frequencies, it becomes highly conductive, probably quantum conductive. He also showed that if you applied 2 megahertz to a bunch of tubulins and measured how long they were, that at 2 megahertz they grow the longest. So 2 megahertz stimulates microtubule assembly. He then turned to neurons and put nanoprobes, STM tips, inside neurons. And he did this at MIT. And he applied them to a whole neuron, to individual microtubules, and the tubulins, and got these interesting triplet patterns, he called them, that are at different scales that seem to be self-similar. So a lot of people think the brain is like a fractal, where information repeats at different scales. And I think that's probably true. And he's shown this, but those people talk about from the neuron up. But he took it from the neuron down. And basically, if you put all that together, what it looks like is, if you look inside a neuron, you get a microtubule, and then a tubulin, and then the dipoles, and you see these self-similar triplet patterns at each scale, kind of like a fractal. So he then, let's see if this is going to work. So this is he then made a sound recording. So this is taking those triplets and down-converting them into audible frequencies. So this is kind of like the microtubules inside your neurons singing, as Anirban calls it. And it's kind of cool. I don't know what it means exactly, but it's kind of mesmerizing, some people think. So if you speeded this up many, many orders of magnitude, that's what's going on inside your brain. This is baseline, and then when you vary it, that's how you get the dynamics that lead to consciousness. Okay, well, here's the last one. Somebody's taking this and making it visual, and that'll actually be presented at the Tucson conference. So this is the sound of your microtubules singing inside your brain. Much slowed down. You're also much faster than some other things. Okay, so let's just pick one frequency, megahertz. If brain microtubules resonate in megahertz, could mental and cognitive disorders be treated by applying megahertz vibrations to the brain? Now, there's various ways of stimulating the brain non-invasively. Transcranial magnetic stimulation, transcranial electrical stimulation, and also ultrasound. And ultrasound is mechanical vibrations above audible threshold, above 20 kilohertz up to 200 megahertz. It's been used in medicine for almost 100 years for imaging, for example, looking at babies in the uterus. And then this guy, Jamie Tyler at Arizona State, he's now gone on, made a couple companies, started looking at ultrasound effects in the brain of animals, and built the first headset here. He started a couple companies making these devices, and he built these headsets, the U+ ultrasound headset, raised a lot of money, and you can see that if you put these headsets on, you get beautiful and sexy. And then there's me. So I heard about this before I made the headsets, and I told my friends in anesthesia we should try this on our chronic pain patients, and they said, we tried it on ourselves first, and so I volunteered, and you can see my my brain on ultrasound there. And for just you don't feel it; it's completely painless. And when I first did it, I said, "Well, that's not going to work." Put it down. And then after about a minute, I started to feel really good. I got a buzz for about an hour. And so we did further studies. We published this, the first human studies on ultrasound. And then I got the experts, including Jay Sanguinetti, who's sitting there in the third row, and Tyler's still involved, and Ezra Smith, and John Allen, Chris Chan, and some other people. And we've done a number of other studies showing that where this is looking at global affect or mood, that ultrasound for, I think this one was 30 seconds versus 10 minutes was better. And that, now that's putting it here. If you put it at the top of the head, the vertex, it didn't really have much effects on mood, but there were some interesting effects like uncontrolled laughter, increased mindfulness by non-attachment scale, out-of-body experience. And we also showed that 2 megahertz was better than 8 megahertz, at least for a while, when they kind of normalized. And 2 megahertz was better than placebo, putting it at the temporal window, it's called. And John and Jay also did a simulation showing acoustic intensity field, that it does get through the brain, it can be focused any particular area and cross over to the other side. And then a paper in mice earlier this year showed that ultrasound, these mice had Alzheimer's disease, and the ultrasound by itself made them better in terms of their symptoms and their pathology. So we know that Alzheimer's involves microtubules falling apart, among other things, that 2 megahertz stimulates microtubule assembly, and this work from Saviki Gupta shows that ultrasound stabilizes microtubules against destabilization. Okay, I'm going to go back to what I talked about earlier real quick and wrap up, and the question of which came first, consciousness or life. And life apparently started in the primordial soup billions of years ago, 3 to 4 billion years ago, a simmering mix. And in the 50s, Miller and Urey simulated the soup, or a soup, and found organic amphipathic molecules, very much like dopamine and the organic molecules in neurotransmitters, which are based on these pi resonance clouds. These were found in the simulation of the primordial soup. Now, since oil and water don't mix, the oily heads stick together and form nonpolar regions. And this also forms proteins and also membranes and DNA. They're all based on pi resonance. And with terahertz radiation that was in the atmosphere, they would start to oscillate. So we're proposing, not quite, we're getting ready to suggest that back in the primordial soup, these protocells, or micelles they're called, by Operin, who came up with the idea of the primordial soup, coalesced and led to, by the E equals H over T mechanism, primitive consciousness. And some of these would be random, but some might be positive and feel good. And this would feed back on pi stack arrangements, and life would organize along those lines. Now everybody says, well, genetic survival, blah, 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 but it was a long time from the primordial soup till we had DNA. So how did life get from nothing to DNA? So, and even after DNA, this whole idea that the genes survive to, our behavior is oriented towards surviving our genes doesn't really make any sense. Anyway, this also, so that would be primitive, simple consciousness. And then when did orchestrated consciousness arise? And one possibility is here in the what's called the Cambrian evolutionary explosion, when in a brief 10 million years, all the animal phyla emerged suddenly, and including structures like this, actinospherium, and those things sticking out are actually a bunch of microtubules in this helical array. So the suggestion is that these dipoles and microtubules are actually connected to structures, to oscillations in the structure of the universe. I showed this before, and the universe itself probably has multiple layers. It's hierarchical, and so the consciousness might go down into the structure of the universe, maybe more intense as you go. And this relates, and then here's the last slide before the conclusion, that it's called the anthropic principle. This is a very interesting question. It turns out that there are 22 numbers, constants, that govern the universe. The ratio of the mass of the proton to the electron, and et cetera. It's the gravitational constant. And it turns out that they're exactly what they need to be to have stars with light, life, and consciousness. So the universe is somehow perfectly tuned for life and consciousness. And there are two explanations for this. And this is called the anthropic principle. It's really a question. And the weak anthropic principle, put forth by Brandon Carter, is the answer is selection bias. He goes back to the multiple worlds hypothesis. There's a gazillion, infinite number of worlds, and we're in the one and only universe that is tuned for life and consciousness. So we're the ones asking the questions. All those other universes, there's nobody there to ask the question. And that's a hard argument to refute, because who knows if they even exist. But that's one argument. Now, the strong anthropic principle by Barrow and Tipler is that life and consciousness are intrinsic to the universe and driving its evolution. And I think our theory kind of supports this, and especially with Roger's idea of previous eons. So we think consciousness is intrinsic to the universe, and the universe over time, and maybe even over cycles of Big Bang, is changing to optimize consciousness. So the universe is also evolving to optimize consciousness. So, in conclusion, the brain is a multiscalar vibrational hierarchy extending to quantum processes, a quantum orchestra, and consciousness is more like music than computational output. Don't believe all that, brain mapping stuff. The Orco-War theory is supported by evidence, while other theories remain untestable. It's not proven, but we have a lot of testable predictions that are pushing it along the way. And resonating brain microtubules with transcranial ultrasound offers promise for Alzheimer's, traumatic brain injury, and other disorders. And finally, our conference that we have every 2 years in Tucson is coming up. And a lot of the ideas, and many others, it's not just my ideas, but all ideas are welcome. And it's out of Ventana Canyon in April, end of April, and there's some flyers here. You're all welcome, and thank you for your attention.